Hello friends, welcome back to my channel. We are in part 9 of biomolecules and we're going to discuss about nucleic acids today. That's the last part of biomolecules, and we all know that there are two types of two types of nucleic acids, that is DNA and RNA. Okay, D-ribose nucleic acid and ribose nucleic acid. What is the difference between the two? We are going to see that in full detail. Okay, so where can you find the DNA? So DNA is present inside the cell. You zoom into the cell, you'll find the nucleus. Inside the nucleus, if you further zoom in, you can see the chromosomes. These chromosomes are present, you can see them only during cell division. And when you zoom into the chromosomes, you can see super coiled chromatin fibers. If you zoom into the chromatin fibers, you can see something called as a super coiled double helical DNA. Okay, super coiled double helical DNA, which is moving from 3' to 5' and from 5' to 3'. That is, both of them are moving anti-parallel. Okay, both of them are moving anti-parallel. You can imagine the DNA just like a twisted ladder. Okay, you can see a ladder twisted from both the sides, and that will become what? DNA. So if you zoom into the DNA, a segment of the DNA is gene, which is the functional unit of DNA. Okay, so inside the gene, if you zoom into further, right, you will be able to see nucleotide. Okay, and this nucleotide, how to remember this nucleotide? This nucleotide is just like a cream biscuit. Okay, so cream biscuit में क्या होता है? बीच में क्रीम होता है, दो बिस्किट्स होते हैं. वैसे ही इसमें क्या होने वाला है कि बीच में राइबोस शुगर है. क्रीम कैसा रहता है? मीठा रहता है ना, तो शुगर भी मीठा होगा. वो बीच में है, राइबोस शुगर बीच में है, और एक ग्रुप यहाँ पे द लेफ्ट साइड इज अ बिस्किट. विच बिस्किट इज दैट? फॉस्फेट ग्रुप इज द बिस्किट. एंड विच इज द बिस्किट ऑन द राइट हैंड साइड? This is nitrogenous base. Okay, so nitrogenous base can be of 4 different types. In case of DNA, it can be either of the type A, adenine, or thymine, or guanine, or cytosine. If this is RNA, then the nucleotide will just replace thymine to uracil, right? Instead of thymine, we'll have uracil. Remaining else will be the same. AUGC, ATGC. ATGC for DNA, AUGC for RNA, ribose nucleic acid. Now, what are we going to do is we are going to see the detailed structures of all these nitrogenous bases. Then we'll see the structure of the nucleotide, and then we are also going to see the detailed difference between DNA and RNA. So let's see how to remember these structures of adenine, guanine, then thymine and cytosine. Okay, so first thing, adenine. Now I'm teaching this from the basics. Now which ring is this? This ring is the cyclohexane ring. You put one double bond in this, it will become cyclohexene. You put another 2 double bonds, it becomes benzene. Now from benzene, you'll have to attach a nitrogen over here. Okay, you'll have to attach a nitrogen over here, and when you attach that nitrogen, it becomes what? Pyridine. Okay, what is it? Pyridine. Then if you attach 2 nitrogens over here, okay, if you attach 2 nitrogens over here, one in this case and one in this case. See, when I ask most of the children, when you attach a nitrogen, and what does it become? The children say that it is nitrobenzene. It's absolutely wrong, because nitrobenzene is something like this. NO2 attached to benzene ring, that is nitrobenzene. See, nitrogen over here, when it is attached inside the ring. By removing a carbon, we have put in nitrogen. Here we have NO2 attached to the carbon, attached to the ring. Here nitrogen is not attached to the ring. We have replaced the carbon and attached a nitrogen. So this is pyridine, this is pyrimidine. So pyridines are thymine and guanine are the pyrimidine bases. Okay, thymine and cytosine, sorry, not guanine. Thymine and cytosine are pyrimidine bases, and adenine and guanine are purine bases. Let's see what's purine. So for understanding what's purine, you need to see one more structure. Okay, so look at our journey. How have we started? We made a hexagonal ring. That was what? Cyclohexane. We put 3 bonds in between, it became benzene. You put 1 nitrogen in between, it becomes pyridine. You put 2 nitrogens, it becomes pyrimidine. Now take a 5-membered ring. Okay, now 5-membered ring, you put 1 nitrogen in between, it becomes pyrrole. Then you put 1 more nitrogen here. By replacing a carbon, you put in 1 more nitrogen here, it becomes pyrazole. What does it become? Pyrazole. Okay, so this was pyrimidine, this is pyrazole. Now you fuse, I'm not saying join, okay, you fuse pyrazole and pyrimidine together, and when you fuse this pyrazole and pyrimidine together, what structure you will get is the purine. So have a look over here. Nitrogen, yeah, I'm drawing the pyrazole ring. This is your pyrazole ring. This pyrazole ring we are fusing with pyrimidine. So This is the pyrimidine structure. Okay, now check this carbon, these two carbons, okay, are over here and they have become common, which means that this double bond is not only in this ring, it is considered in this ring also. It's common for both the rings. Which one? This double bond is common for both the rings. So therefore what's going to happen is now I'll have to put a double bond over here and over here only, and this becomes the structure of what? Purine. What does this become? Purine. So in this purine, if I simply attach an NH2 group, okay, the moment I attach this NH2 group, it becomes something called as adenine. Okay, this is the structure of the first nitrogenous base which we are seeing, that is adenine. We have come a long way step by step. So when I attach NH2 is adenine. So purine is the nucleus. Yeah, try to understand this. Like if I have chlorobenzene, if I have phenol, and if I have aniline, so if this is aniline, this is chlorobenzene. Okay, then if I have, say for example, bromobenzene or phenol for that matter. So in all the cases, what is the nucleus? The nucleus is the phenyl ring. Phenyl ring. Okay, benzene minus one hydrogen is phenyl. So phenyl ring is common in all the three. So phenyl becomes the nucleus for all the three. Similarly, purine becomes the nucleus for adenine, guanine, and caffeine. Yes, you have heard that right. Caffeine is present in coffee. Okay, so in coffee also you are having the purine nucleus. So you attach something else instead of NH2, it will become guanine, and you further modify it, it will become caffeine. But the nucleus remains the same. Purine will remain the same. So now if I want to convert this adenine into guanine, how do I do that? Okay, just remove this NH2 from here and attach that NH2 over here. Okay, attach that NH2 at this position and replace that NH2 with a C double bond O. So where's the C? C is in the ring itself. C double bond O. Now you must be observing what's the problem here. Now carbon is having a valency of 4, and here we are seeing 1, 2, 3, 4. 5 bonds are there. So instead of 5, we need only 4 bonds. So I'll remove this double bond. Now we are having 1, 2, 3, 4 bonds. But have a look at the valency of nitrogen. Meanwhile, the valency of nitrogen has got disturbed. It's 1 and 2. So I'll have to attach a hydrogen over here in order to satisfy the valency. So only just play with the valencies and you will get the structure of guanine. Okay, so what did we do from adenine? For converting adenine to guanine, we replaced this NH2. I mean, this NH2 we removed and attached over here, and in place of NH2 we put the C double bond O and just adjusted the valencies. And once we did that, we got what? Guanine. So guanine is also a purine nucleus. Okay, guanine, adenine, and caffeine, these three are having the purine nucleus. So we have done 2 nitrogenous bases so far. Now we are going to move on towards the pyrimidine bases. That means they are also nitrogenous bases, but they are having pyrimidine as their nucleus. Okay, so first one we'll start with uracil over here. Okay, uracil. We're going to start with uracil. Okay, so uracil is something like this. Have a look. You will be having, it's a pyrimidine nucleus, so it is but obvious that you will be having one nitrogen over here, and you will be having one nitrogen over here. Okay, then only it will become a pyrimidine, and we have an hydrogen because this is carbon. 1, 2, 3, 4 is the valency. I need to attach a hydrogen in order to satisfy the valency. There is one carbon here and there is one carbon here. So what are we going to do is we are going to attach a double bond O and a double bond O in both the places. And this becomes the structure of uracil. See, now if you remember the structure of uracil, the other two are free. That means if you remember the structure of uracil, you will be able to automatically remember the structure of thymine and cytosine. So uracil is this. Okay, you need to attach hydrogens in order to satisfy the valency of the nitrogens. Okay, so here this is the structure of uracil. So if you attach a methyl group to the uracil, in other words, methyl uracil, okay, I repeat, methyl uracil is nothing but thymine. Methyl uracil is thymine. That means this is your uracil group. If you attach a methyl group over here, the moment you attach a methyl group over here, it will become thymine. Now it's no more called as uracil. It will become what? Thymine. So how to convert uracil to thymine? Just attach a methyl group. It will become what? Thymine. Okay, so now coming back to the structure of uracil, because we want to learn the structure of cytosine now. So again we fall back to the structure of uracil. What was uracil? It was something like this. Carbons over here. Then we are having a C double bond over here, C double bond over here, nitrogens in here with one hydrogens attached to both the nitrogens. Okay, so this is the structure of uracil. Now I want to convert uracil to cytosine. So what I have to do for converting it into cytosine. So remove this C double bond O and attach an NH2 over here. The moment you remove the C double bond O, attach an NH2 and just satisfy the valency of the carbon and nitrogen by putting a double bond over here, baat khatam ho gayi. This becomes what? Cytosine. So what did we do to convert uracil to cytosine? We replaced the C double bond O with NH2 and adjusted the valencies by removing the hydrogen from here, and we got what? Cytosine. Very simple. We got what? Cytosine. So we have remembered uracil, then thymine, and finally cytosine. So these three nitrogenous bases were pyrimidine bases, and the first two, adenine and guanine, were purine bases. So we have covered all 5 nitrogenous bases. Now let us see the structure of a nucleotide. So nucleotide will be having, I told you, nucleotide is like a cream biscuit. So in between we'll be having a ribose sugar, then phosphate group. So let me draw the ribose sugar over here. Okay, I'll just draw the schematic diagram of ribose sugar at the moment. Okay, so this is your ribose sugar. So if this is the first carbon, second carbon, the second carbon, I mean, usually it has an H and OH group attached over here. Okay, but in case of DNA, this is going to be, instead of H and OH, it's going to be H and H. That means it's going to be devoid of oxygen. Devoid means without oxygen. And because it is without oxygen, it is called as, DNA full form is deoxy. D meaning removal of what? Oxygen. Oxygen from where? The second carbon. So you remove the oxygen from the second carbon, okay, and then it becomes what? Deoxy ribose nucleic acid. Ribose is there, deoxy where? On the ribose you remove the oxygen on the present on the second carbon, it becomes deoxy ribose nucleic acid. Okay, then this is the ribose. We are going to connect what? A phosphate group over here. Okay, so phosphate group being connected over here, this is phosphate group, ribose sugar, and then you can attach any nitrogenous base over here. So if you attach, like, say for example, the adenine group over here, if you attach the adenine group, you know what is the adenine group, right? So what is the adenine group? Yeah, where is the adenine? This is your guanine. Okay, even you can attach guanine also. Guanine or adenine, if you attach, then that will become guanine, adenine, or you can attach uracil, thymine, or cytosine. Anything you can attach. Okay, for making it a what? Nucleotide. So adenine, either adenine, guanine, cytosine, or thymine, any of these 4 you can attach over here to make it what? A nucleotide. So the moment you attach phosphate group, ribose sugar, and nucleotide together, it is going to adenine together, it is going to form a nucleotide. Okay, and now if you remove the phosphate group, if you take the phosphate group aside, if you remove the phosphate group aside, this will now become nucleoside. What is nucleoside? Taking the phosphate group aside makes it nucleoside. Nucleoside is ribose sugar along with the nitrogenous base. Ribose sugar along with the nitrogenous base is called as nucleoside, and like this is called as, instead of adenine, now it will be called as adenosine. So what is the adenosine formula? Adenosine is nothing but adenine plus ribose. What it is? Adenine plus ribose is adenosine. Adenine plus ribose is adenosine, and it is called as a nucleoside. Now quickly see the difference between DNA and RNA, and then we'll be done with nucleotides. Okay, so what is the difference between DNA and RNA? So DNA is the genetic material, RNA is involved in protein synthesis. This is genetic material involved in what? Protein synthesis. Okay, for protein synthesis it requires 3 types of RNA, that is mRNA, then rRNA, and tRNA. So these are the 3 types of RNA which are required for protein synthesis. Okay, whereas DNA, it does not involve any you can say it is not involved directly in protein synthesis, forming what? Genetic material. It's not having any types also. Okay, then moreover, DNA is a double-stranded helical structure. What it is? It is a double-stranded helical structure, and RNA is generally single-stranded. Okay, RNA is what? Single-stranded. Then another difference between DNA and RNA is the second carbon of ribose on DNA is not having the oxygen, and the second carbon on ribose of RNA is having an oxygen. So this is deoxy, this is oxy. Okay, then another point of difference will be that DNA is having phosphodiester linkage. So what is this phosphodiester linkage? Now what is phosphodiester linkage? Phosphodiester linkage is the vertical. Now where is the DNA structure which we had drawn? Where has it gone? Yeah, here it is. Okay, so here we can see that these vertical bonds, okay, these vertical bonds are the phosphodiester bonds. Okay, and these horizontal bonds, H for horizontal, H for hydrogen, these are the hydrogen bonds. Okay, so phosphodiester linkage and hydrogen bonds. This is also present in RNA because sometimes mRNA is like single stranded, but this is the hairpin model. It's a single strand, but sometimes it can form the hairpin model and form the hydrogen bonds as well as the phosphodiester bonds. So this is all about nucleotides and nucleic acids.